Very good afternoon to all of you. Welcome back for what is going to be the last and final session, not only of the day, but the meeting, which has been a fantastic one. I think we have had some amazing talks already. We have the final 3 talks of the day. The speaker before the lunch was talking about, the worst time to give a talk is just before lunch, but I think the time after lunch would give a good competition to that. So, we have a tough job at our hand, but, I'm sure we'll have fun discussion. As usual, it will be 30 minutes, 25 plus 5. Around 23 minutes, I'll just get up and show my hand. And I have a request that yesterday we were following the scheme that we would hold on to the questions until the speaker is done with his or her presentation. And I request we stick to that, because I think otherwise it affects the dynamics, and then one question leads to another, and then the speaker has to in the end rush with the data, which is not a good thing. So we'll let the speaker finish, and then of course in the end, depending on how much time we have, we'll take the questions. All right, so with that, let's begin. The first speaker of the day is Avik Samanta from IIT Kharagpur, and he'll be telling us about sculpting life-inspired soft matter systems by harnessing biomacromolecular phase separation.
Okay. Thank you for the introduction, and also thank you for the invitation, because when I first got the invitation, I was a bit shocked why an organic chemist like me, why I'm invited into this conference. But I think last 2 days, or yesterday and today, I was intrigued by all the talks, and it was a fantastic experience. But anyway, so as I said, that I'm an organic chemist by training, and my group in IIT Kharagpur are actually looking at some of the phase separation behavior, not to just about the sake of seeing the phase separation, but also to use that to make different kind of bottom-up approaches to make materials. I'm gonna talk about quickly what we actually and where part of chemistry and science we belong. So normally our group actually looking into dynamic adaptive materials, and these dynamic adaptive materials, they are slightly different than the bio-inspired materials. And bio-inspired materials are normally what as you can see, that hydrophobic surfaces that you can construct by getting inspiration from biology or biological materials, but in that case they are merely static in sense. Okay, but on the other hand, you see that the other side of the spectrum, what we see actually living materials. Living materials mean we actually try to mimic biology in a sense of try to make microtubules, which is a fascinating self-assembly process which goes through the assembly of proteins in out of equilibrium system, which is also fuel driven. And that's what I think we got very excited, where we can actually mimic them from something which you can make in the lab. Also something like cell division, or having that's some change in the macroscopic level when embryo develops. So the whole idea is that we move from the static materials to life-like materials or adaptive materials. They are mostly energy dissipation, active regulatory networks, and system integrations are integrated into the material. So that is something that we are excited about, to make different kind of bottom-up approach materials. So that's why our motto mostly goes to count, organize, move, and adapt. So when we talk about life-inspiring materials, the most important or the first thing that we want to look at is a living cell, and living cell is an inspiration for any chemist to actually understand, or not to understand, not even a small miniature version of this cell with a one process that can actually takes a biochemical or chemical signals and gives a proper function in an autonomous way. That would be fantastic to even think of. So as we call it, sensor, processor, actor pathways. Most of the cases, this signaling network is very complicated in cells, but in our lab, we try to understand whether we can actually form something which is much more miniature, much more simple. So metabolic soft matter system. Anyway, so this is not something that a lot of people are doing. So it's not only us. Many people have been trying to look at compartments from a very primitive way. You know, phospholipids, brochopolymers, as well as some fancy molecules like protein polymer conjugates. These actually forms membranes and it's kind of enclosed a aqueous compartment, and then you can actually put your stuff and you need to see what happens inside, and there's a definitely you need to have a transport phenomenon from the external environment. But anyway, so most of the cases this is a liquid phase separation conference. So one thing that I would like to point out that living cells are not aqueous compartment, they are filled with macromolecules. So they are more like a crowded soup of molecules and they are bumping into each other. So in a very interestingly, Adrian Elock said in this paper, there is order in the chaos. So it's kind of fascinating that even in a very macrometer size pocket of a micro reactor, you can think of how things are actually working. But anyway, so as a chemist, we are interested in looking into molecules. One of the molecules in our lab that we work on is DNA, DNA double-strand helix structure. So this is very nice. But also, we are more interested in single-stranded DNA because these are actually more like a flexible polymer with negatively charged in their backbone, and we want to see whether they have interactions with the divalent cations, and then is it possible to even synthesize a crowded confinement that we have been talking about. So it goes back to my postdoc time, 2000, it's almost 6 years ago. We accidentally found something. So we were working on making hydrogel with DNA polymers, but suddenly because of some change in the procedure, we found that this poly A, so poly A is like homopolymer of A, and we heated up this solution in presence of magnesium, and we found that there is a phase separation at high temperature. So that is like more like a LCST behavior, which is like polynipom kind of, and this only happens, so suppose if you see it here, 400, you can see the turbidity increases in the solution. And this is actually got us quite intrigued because this is also reversible. So you cool down, your solution becomes clear again. So that means the globular form of the poly A goes to solution. But this doesn't happen in the case of poly G. It kind of phase separates in the same way because they're all purine bases, so you have aromatic, larger aromatic surfaces, but they actually get stuck in this globule form when at high temperature but doesn't come back. But this kind of behavior is not seen in poly T and poly C. This is quite interesting because it gives us an understanding that why this purine-based polymer kind of gives us this phase separation behavior at high temperature. So of course this depends on the molecular weight of the polymer. So once you increase the polymer weight, the LCST temperature, there's a sharp point where flexible change goes into globule form, and that actually goes down with the molecular weight. And also if you increase the calcium and magnesium concentration, you see at certain level the temperature goes down and it gets a plateau. It's quite interesting to see that it's some kind of electrostatic interactions and then this kind of charge neutralization. But LCST is quite difficult even to understand from the Fleury-Huygens theory. Anyway, so this is something that we did in under the microscope. We heat the solution, and this is a poly A solution, and then we see like a popcorn kind of mechanism, so nucleus growth, and there's also liquid-like behavior. I don't want to stress that, but it's quite interesting. So now the question is that we wanted to make this crowded environment, but then we got a phase separation in poly A, but then these are reversible. So of course we need to somehow arrest them, right? to kind of really get into this compartment that we have been talking about. What we try to do here is called, like we try to combine these two phenomena. One is the polymer properties of poly A, which is the LCST behavior, and then you have the supramolecular interactions, which is more like a duplex formation, because it's a DNA. So what we have done, interestingly, we took two polymers, and this is actually a bit different than homopolymers because they have repeating units, and these are actually can be synthesized by RCS, or rolling circle amplification, from a template, circular template, and we synthesize two polymers where you have repeating unit A20 and M. M is just a barcode you can think of. It's a sequence that we just amplified from the circular template. But same way, we synthesize a T20N polymer, which is orthogonal. This sequence, blue one orthogonal to orange, and green one is A20, red one is T20. So when you mix them together, of course we'll see A20, T20 interactions in solution, but what we have done, you kind of quickly heat it up to 95 degrees. Now we're talking about two temperatures. The one temperature is the cloud point temperature of the poly A, which is the green polymer, and that we know from the last slides. So we added magnesium 50 millimolar, and then this one doesn't phase separate, we know that. But there's also a temperature called A20, T20 duplex formation, which is slightly higher, the melting temperature, than the TCP. So when we cool down, we go to the high temperature, we kind of see this, the globule formation of the green polymer, and the red polymer stays in the solution, and when it quickly cools down, what happens that the red polymer wraps around in the droplet through and the circumference of the droplet and the surface, and forming this A20, T20 duplexes, and then it's kind of get arrested, the droplet. So now, once you kind of cools down, this droplet wants to go back to solution, but because of the formation of this duplex, you kind of arrest this. So this is more like a kinetically trapping of liquid-liquid phase separation droplets. So once we have that, and you have this code barcode and the shell barcode, that is actually the identical of M and N respectively, and we can actually use a die which has this N star and N star, and these dies can actually go and orthogonally go attach there. So the green die goes to the core of the particles, and the red die goes to the surface of the particle. So this is very interesting because you have a shell and you have a core which is full of liquid DNA, and of course you can see the higher the temperature you go, the structures are getting more defined. So we do FRAP, and then we see that the inside of the particles, they are quite liquid, so if you bleach half of it, but the other side, the shell is more like a hydrogel, so they're very cross-linked. One more thing that we noticed that once we have the only single standard domains, so the poly A, and then if we increase the duplexes inside by just hybridizing the M with the M star, that the polymer, the whole particle swells. And this is because the persistent length of the duplex is longer than the single standard. So, so what happens that it's kind of gets very difficult for the droplet to stay in this phase separated form. So it gets much more liquid. But anyway, we did a lot of interesting structure formation by just playing with the LCST of the polymer and also the hybridization of the DNA strands. So this is quite interesting where we have gold nanoparticles, and then we can actually use that to pop the shell by using a 561 laser. So I quickly, this is even a bit old, but still I want to show you that as a chemist we wanted to understand whether this kind of structures or this kind of microscopic particles can be used to do chemical reactions inside. And then if the enzymatic activity in the we can call it intraprotocellular environment, if we call them protocells. So that's how the activity of the enzymes get affected by the crowded environment of the DNA. So this is something we were really interested, but at the end what we wanted to do, chemical signal get processed, and then we have some kind of downstream signal propagation, either like a tissue kind of structure formation or functional adaptation. So one quick example, this is a catalyst that we synthesize. This is called Grab's catalyst. It's do a reactions which close the rings and release this fluorophore. So you kind of think of as a pro-fluorophore. And this is actually sitting inside a streptavidin pocket because there's a biotin attached to the ruthenium complex, which is the catalyst. And then we can actually change the mutate the enzyme to see whether the activity of the enzyme gets changed in the crowded environment. And we kind of call this as a directed evolution. But anyway, not so important in this talk, but what we wanted to see that the DNA environment has an effect on this artificial metal enzyme. And we have seen that some of the mutation, even though it's very difficult to rationalize those results, but in the screening process that we have done almost 80 proteins, and then we have seen some of them almost increase 5 times activity. So we have also seen that the specific reactions kind of generate a strain in the particles, and also there is a fusion effect that kind of change the structure of the particles. Also there is a interdigitation of the membrane as well as fusion. And one of the other examples where we have a DNA zyme which kind of catalyze the breaking of a RNA bonds in DNA. So that's actually also was used inside because the enzymes can be easily incorporated by using these barcode interactions just to and then you can wash these particles. So there's no excess catalyst floating in the solution. So here also we have seen that the specific quenched substrate can go inside and the catalysis can happen, and you see a signal which is changing over time. There's also a certain change. So like once we do the cleaving of a specific substrate, and then if it dissolves, that substrate can also comes back to the surface and change the color, as well as expose a palindromic sequence which kind of cross-link the particles. So this here we are actually doing a metabolic reaction, catalytic reactions, and then because of the reaction the particles changes its color as well as try to find each other by duplex interactions. And this is quite interesting because it shows how a completely inanimate object can actually gives you a structuration where you just add a chemical signals and then the particles kind of change itself and then try to find each other using a tissue kind of structures. But anyway, so very quickly I'll show one more important. So this is not published, and this is actually we're trying to do in my group. What we're trying to understand whether since we have a DNA a crowded environment, which is more like a liquid-like DNA, and then we can also change the mechanical properties because we can also cross-link the particles, the core of the particles, by using DNA, different kind of DNA tools. But also we want to see whether any kind of phase separation of the proteins, which is actually known like a fast protein. This is actually a disordered protein, and we can actually want to know whether this kind of proteins can be phase separated and inside, and we can actually see whether the structuration and also various properties can be tuned by the DNA interior. And this is something we are working on. This is a sticker, a special model, but the sticker is light responsive. So every time you switch on a light, you can actually go for back and forth with different phase separated and liquid particles. Okay, so fast, I think I don't want to go into it because this is very well known. Fast people have been working on this. So the fast actually gets phase separated in this paper, Dorothea Dorman, and she showed that if we change, mutate this NLS region, the fast get phase separated at the cytoplasm region, so that in the nucleus normally fast is well soluble. And this is working, the fast has a lot of activity and functions for the DNA splicing and different kind of factors. And once you have these kind of things, you also see ALS and neurodegenerative diseases. So on the fast droplets, they also found that over time the fast droplets kind of go into solidified so it's side of percolation, so like liquid goes to more like a solid-like structure over time. Anyway, Johan and Dorothy, they have been working on this for a long time, so I asked Dorothy that whether they can give me some of their protein. So the protein also have an MVP tag and a GFP tag, and of course it's very interesting that we can actually follow the phase separation. And as you can see from their data in solution, once they have a TEV protease, which is cleave the MVP tag and the phase, the fast phase separates. So what we wanted to do to bring the fast protein inside our particles, and because of that, so we have a very high concentrated protein solution which is mixed with DNA, and now once we have the TEV protease activity kind of get kicked in, and that's why we kind of see whether fast will show any kind of phase separation in the presence of crowded environment of DNA. So that's what we have seen. Quite interestingly, we see that a droplet-like structure formation, and fascinatingly, these structures are quite stable, and we have not seen many fusion, so they are kind of stabilized in this concentration of protein. And so they definitely want to see whether we can actually change the concentration of the protein, and we can actually achieve different kind of structuration inside the particles. And once we have done that, so this is actually fast GFP divided by the repeating unit concentration of the polymer. So because we cannot change the concentration of the DNA in the particles too much. So that is why we kind of try to see whether the ratio changes, and we can actually see from the droplet-like structure. So these are all taken within 1 hour of the experiment. So we didn't age this sample. So all these things you can see that the droplets also it can go slightly spinodal region, but here also once these domains kind of fuse together and then wanted to go to the membrane and weight the membrane and forms these vacuole-like structures, and at some point of time you will not be able to see any phase separation because there's exceeding the loading capacity of the particles. We also see that the DNA which are present in the particles, and if we color them, so during the RCA when we make the polymer we just covalently attach a dye, and the red dye we see that the DNA and the color of the fast is actually co-localizing, so that means DNA is also getting pulled into the fast droplets, which is quite interesting to even think of. Also, as you can see, we have done some of the aging experiment, and this concentration where we started to see some kind of spinodal droplet, and then we see the weighting of the droplet or the membrane over time once we don't have any duplex domains inside the particles. This is M, this is A20, and once we have a duplex domain, this kind of phase separation is bit changing because the duplex, the double-stranded DNA, they don't really fast don't like them. So they don't want to get incorporated inside the particles because of the problem with their phase separation behavior. So it's more like antagonistic behavior. And then you see there are a lot of droplets are forming outside the particles. And once you kind of see there's an RNA, which is known from the previous studies, that RNA, particular sequence of RNA is called SON RNA, and that is actually binds to fast even stronger. So even if you have duplex domains, you can increase the RNA sequence inside, install it, and you can also see the phase separation again increases, and propensity of the phase separation increases. So once we did this kind of mobility assay, we have seen that A20 is the very bad binder for the fast, but M has a quite high binding affinity, but also M star RNA plus, which is definitely the domain of the barcode that we already have, the complementary of it, and the RNA plus, which is known already, they have a very high binding with the fast. And that is also we have seen, we increase the concentration of RNA, we also see over time the soluble part of the so it is kind of early phase separation by just introducing the RNA, even though there is a 22 place, has a very low affinity towards the fast. Yeah, so one more thing that I wanted to show you, that if we cross-link the DNA inside, so now we are changing the mechanical property of the core, and that actually has a huge effect because now the fast is actually, this is first MVP before we added TEV, and once we started doing this, this is 0%, so you have seen already the phase separation behavior, but once we increase the cross-linking, we kind of suppress the phase separation behavior. Yeah. So this is something quite interesting because as the mechanical property changing, the core is less liquid. Now the first phase separation is suppressed, which kind of brings us to one very interesting experiment. Once we have the cross-link particle, there's no phase separation. After 4 days old sample, we added RNA and we started to see the phase separation outside the particle. So the first is kind of moving from, which is a disfavorable situation, from the one where they can bind the RNA outside. So same as well, if we increase the duplex concentration in the shell, normally they like the single stranded DNA and they kind of populate in the surface, but once we increase the duplex DNA concentration at the surface, it goes homogeneous. And one more important thing that we have done, this is actually the particles that we A20 and M, there is no duplex domains, and then once we phase separate them with fast, and then if we swell them, we remember we increase the duplex domain inside, the particle swells, and that's why, but because this is already aging, so the protein droplet which is formed, which is kind of weighted the surface, now that kind of solidifies, and then once you add DNA zyme, you kind of get this fast capsule-like structure. So anyway, so I think this is where I'm going to stop because I don't want to, I think we are also over the time. Something that I wanted to show, but I think because of the time I could not. So this is something that I talked about, how we can change the spacer sticker model by using a light. So once you increase the concentration of the green the small molecule, which kind of use as a sticker, but the red one doesn't, and we can also see that we can change the phase separation behavior of the DNA inside by using the particular light. We can also go the spinal decomposition, which kind of also goes to the wall, internal wall, and weight the particles. And then depending on the magnesium concentration, this can also have a different structuration. So once we cross-link, we can get a network-like structures where this is kind of doesn't go to the wall, but we can actually go back and form because of the light changing. But anyway, so I think I will take these two questions because I think there's some confusion. Please ask me. So what we're trying to do, I think, is actually try to make microparticles which looks like a living cell, but we also, it's a very minimalistic model to understand liquid-liquid phase separation in a crowded environment in a very controlled fashion. So if you have any questions, please let me know. I'll be very happy to answer. Yeah, so this is my group in IIT Kharagpur. Johan has done a lot of the protein synthesis in mines. Yeah, thank you very much.
Thank you, Avik, for finishing on time. We have full 5 minutes for discussion, so go for it. Yeah.
So Avik, so you showed some slide where you said, the poly G does not phase separate temperature and poly A does. So I'm wondering if that is because in poly G you have this possibility of quadruplet.
Yes. So it does phase separate, but it doesn't come back to the solution again.
So in poly A, is it possible that the mechanism is where you are forming some interdigitated stacks between different strands?
But during the phase separation process,
yeah,
yes, yeah. So we haven't built it because we wanted to see some experiment in a molecular level to understand how this is forming. But definitely there is a stacking formation.
Yeah,
so this is aromatic.
Why I'm asking, because we did some simulations of poly A.
Okay.
Where we wanted to look at what is happening at high temperatures.
Yeah.
And what we find which was surprising is, at low temperature you see this intra-strand stacks, sort of breaks, and high temperature it sort of interdigitates between different strands.
Okay.
I was wondering if that could be one of the possibilities.
That could be, but I think still we will not be able to see that in the experimental setup. But there are some recent paper from Priya Banerjee's group where they are looking at the RNA part of the polymer. So they also see this kind of similar behavior, LCST.
Yeah.
Yes.
Yes, I'm curious about the wetting behavior. So you see wetting inside the structure and also on top, like on the surface of the. So,
Yeah.
so if you go back, some of these. So I'm more interested in on the ones that you form on the top, like on the actual surface of the things.
So
You actually you see, right, there is wetting on the surface.
Yeah.
So there, can you measure the angle and really characterize the...
Just a contact angle, yeah. So that is we haven't done yet, but I think this is going to be one of the, but I don't know how valid this would be because this is more like in a three-dimensional and depending on the...
It's possible to do that. I mean, there has been some attempts. You can still do it. You
Okay,
don't
okay.
Curved structure.
Curved,
But
okay.
the question is that what is your tuning now? Like what can you change the temperature? What are the things that you can control? I mean, because can you change the temperature or the interaction of these molecules to the surface?
Yeah. So what we have seen that when the fuzz is kind of loaded into the particles before the phase separation, but they have a lot of duplex domain, so the polymer is much more stiff, and then we have seen that most of the cases when you add RNA, which is kind of sitting at the surface, fuzz kind of likes to go back and finds their own RNA and kind of phase separate at the surface. So this is the tuning so far we have done, but we haven't done any change of the metal ions and also the temperature, because I think fast phase separation, I'm not really sure how the temperature behavior, because I know if you go to low temperature, I think this will the affinity of the fast interaction will increase, but I think we haven't tried this, but I think this is going to be one of the ways to tune the properties.
I have a question.
Yeah.
I'm an experimental biologist, so I'm sorry if it comes across to you as why am I asking this. So this looks fantastic. I'm just trying to understand, your aim is not probably to understand the disease better, right? I mean, FUS is just happens to be a protein that you used in your system. Is that right?
Yes. So what the main idea when we started this is that how does the phase separation would happen in a confined state? Like, is this kind of some kind of structuration that we see in solution will be still accessible, or we will see, because at the end what it's doing is even the concentration is increasing, right? In local concentration is increasing, but we haven't looked into it. But then the question also always asks that how fast is get solubilized in the nucleus, but not in the cytoplasm, even though there's this very small mutation in the FAS. And then our model system is also a very high concentration of DNA concentrate or condensate. So that means there is some kind of resemblance. We can put, even though this model is still very primitive level because it's only A20 and specific, but if you increase the concentration of genes or the histone proteins, will this be a mimic of a nucleus? And
okay, so you do want to now address
yes,
the
that
biology
is
of the disease using this as a system now? Is that?
I don't want to say that, yes, this is, I'm thinking that this will give us some insight that cannot be done in the living cell because this is going to be a model system.
Okay, so then my follow-up question is that, nuclear condensates of FUS are also known, right? Which is what at some concentration of DNA and RNA you see it's happening here, right?
Yeah.
So this kind of a confined system, what additional information does it provide? So for example, ambiguity-tagged in vitro sedimentation assays that Dorothy does and other people do are well standardized. Here, what is the new thing that we can learn using this kind of a system?
So far we are looking at the properties of the FUS, not even like changing, like not trying to correlate what we have in the nucleus. Of course, there are many factors in the nucleus that is difficult to recreate in this synthetic model. But what our goal is to kind of see that the mechanical properties and the crowding and the hydrodynamic confinement, how we can actually slowly understand that even this is a model protein, but how we can actually understand the phase separation behavior, as well as the idea would be to actually go into more active proteins. So if we can do a reactions which kind of form in situ phase separation, is these reactions going to be enhanced once we have the droplet inside these particles?
So you might want to take mutants in the next step and see how they behave.
Yeah.
All right. Wonderful, fantastic. Yes, please.
fascinating one. So I have questions, like you showed spinodal decomposition as well. So how do you characterize spinodal decomposition as compared to binodal? Because it has a very different property, like it is a nucleation limited phenomena. So how do you characterize that? Like it's based on like structure, morphology you are observing, or there is other way to characterize it?
Yes, so I think so the nucleation and the growth process is actually a bit different than once we see in the confinement, because the timeline is different. Sometimes it get very delayed because of the diffusion coefficient of the molecules that's different in the confinement. So come to your questions, yes, we didn't do any simulation, or couldn't do any phase diagram, because the concentration of the DNA inside the particles is very difficult to change for us. We can alter some, but it's very difficult to change in a very systematic way. But we are looking at the particles from the microscope, and we're trying to understand the concentration of protein where actually this specific droplet goes to the spinodal decomposition.
So like how to characterize a spinodal decomposition, like from the imaging, how do you know that this is a spinodal decomposition as compared to it's not like phase separation going like?
So you mean saying that the because spinodal decomposition, so you have the metastable state.
Like
Yeah, so you have the two phase of the diagram. So you have if you see that there is and there's a little bit of fluctuation kind of change the goes to the spinodal. So the spinodal, you can see the concept change in the microscope. So you have concentration increasing, you have but the spinodal is also, because of the concentration dependent, also you might not see that the droplet forms and then droplet goes fuse and go to the spinodal. That we don't see it, but we can directly jump to the spinodal decomposition, because that is the concentration that approves that. So it's a more visualizing the kinetics of it. I hope I answered the question.
yes.
All right, let's thank Avik for a wonderful talk, and we now move on to the next speaker, that is Snigdha Thakur from ISET Bhopal. She'll be talking to us about dynamics of flexible active polymers.
Let me start by thanking the chair and the organizers for having me here. My apologies, I am not going to really talk about biomolecular condensate. I'll definitely talk about the condensates, and towards the end maybe I'll try to say that the model that we have developed in the context of some other synthetic polymers can very well be applied to the biopolymers. That's the argument that I will try to convince you towards the end of the talk. So I'm going to talk about the flexible active polymer, And to give you idea of active systems. So far we have talked about this condensation formation, this coil to globule transition in the context of electrostatics most of the time. We had polymers which were mostly charged, and the charge was playing a very crucial role in forming these transitions from the coil to globule state. That is what we have seen so far in many talks. What I'll try to convey to you that charge is definitely one way of forming the condensate, but there are other phenomena, there are other forces that can also be very relevant in forming these kind of globularization of the polymer. Right, so before going to the polymer, what are the active systems? Active systems are something which has some internal agents to generate motion, to generate some mechanical work by consuming the energy which is there in the environment. They don't need any external forces for it to move or to perform certain work. They have some internal mechanisms by which they can perform the mechanical motions or self-propulsion. Bacteria is one example for that. The natural motors that we have at the nanoscales are other examples of that, and we have examples at different length scales for the active systems. Birds, humans, fishes, all of them are active systems. So starting from nanometer all the way till our scale, we have active systems present in it. And the interesting part of these active systems are they exhibit a very fascinating collective motion. So you can see this flocking of birds, fishing, the school fishes, the way humans behave in a herd, the way bacteria swarms in collection. These are very fascinating collective behavior which comes in these active systems automatically, irrespective of the length scales in which they are. Right? So active agents are the agents which are employing some energy from the environment and giving some forces to the system. Right? This is one example in nature which is active polymer, which can be classified as active polymer. And what do we mean by active polymers? So you have chromatin. Chromatin has all these enzymes, proteins, which are doing a lot of different types of work, and therefore they are giving some forces locally, some forces to these polymer, and hence it makes it an active polymer. And it has been shown by these people that these active processes are different at different parts of the chromatin. For example, euchromatin, which is more open, has more activity than heterochromatin, which is more closed, right? And also they have very well showed that these active processes, when they are correlated, so if you have multiple active processes happening on the chromatin, right? By active processes I mean these enzymes, these transcription factors, they are all working on the enzymes at different locations, but if they are correlated in the way they are working on, this can actually mediate some effective attraction between the polymer, and that correlated activity can also lead to some sort of a compartmentalization in the active polymer. So what I'm trying to say that these active processes, these active forces which are there locally at different on the polymer can also give rise to some sort of a condensation by this effective attraction. Okay. But that's not what I am trying to talk about right now. I am going to talk to you about the active polymer, which are synthetic active polymer, and we'll try to take it from here to the natural active polymer also. Now, what kind of synthetic active particle that I am talking about? I am talking about this kind of Janus particle, which has two faces. You can see there is a red face and a blue face, and they are chemically very different from each other. Right? The red face, which is platinum coated on a silica sphere, which is the complete sphere, carries out a reaction on just one side of the Janus, and because of this reaction, what happens is there is a gradient which is formed of the product or the fuel, whatever you want to say, and that gradient somehow propels this Janus particle in a particular direction, right? So this is a particle which does not require any external forces for it to move. It has the mechanism, right? It has some mechanism by which it creates the gradient for itself to move. Right? Therefore, it's an active particle, it's a self-propelled particle. Right? Now, if I have multiple of them, right, and this has been characterized well in the literature, the single active particles. Now, if there are multiple of them and if we stitch them together by some means, okay, there are different means by which people have stitched these active particles, they form these polymers. Right? And it's been shown that these polymers exhibit very fascinating, definitely the motion. If you stick them like this, leave them, These ring polymers, they will show rotational motion, the linear one will show a directional motion. If you clamp them, they will beat like cilia, and so on and so forth. So this is something which is very fascinating from the perspective of synthesizing something which is natural also. Okay? So this is one of my main motivations for my model. But then I would just like to give you a glimpse of what other type of active polymers are there. Synthetic active polymers are there, which is in the literature. You can also see these. These are the polymers, passive polymers, which are in the active bath. And they show very nice enhancement in their dynamics. And similarly, this is again ice templating of, you have these active particles which are stitched by some other mechanism, and that they also show a very enhanced diffusion and so on and so forth. Okay. Now to understand these stitched Janus particle, okay, there have been different models. So to understand this active polymer, there are different types of models which are there in the literature. One model which is called active Brownian polymer, where what you have is the polymer chain, okay, and these activity, when I say activity, it means some sort of forcing, right, which is there. The origin of forces could be very different for different systems, but you have some forces, and these are represented by these these red arrows or these red balls here, and they are completely Brownian. That means the monomer of the polymers will have forces in all the directions, right, and that's why they are called active Brownian polymer, and they exhibit this kind of the reduction in Rg and then the swelling behavior. So the non-monotonicity in the Rg is observed for this kind of active Brownian polymer. There's another class of active polymer which people talk about in literature, which is called polar active filament, right. These are the filament where you have forces on these monomers, but they are not Brownian anymore. Rather, what you have is the forces which are directed along the backbone. So if I'm sitting on this monomer will have a force along this direction, this monomer will have force along this direction. So there is a force which is polar in nature along the backbone of the polymer, and this is why they are called polar active filament, right. And this has some relevance in the biology. I'll come back to that towards the end of the talk. And what these people have shown, that if you have such polar active polymer, then depending on the activity, which is described by this number called Peclet number, larger is the Peclet number, more is the activity, depending on what Peclet number you are working on. As you increase the activity, the polymer shows a coil to globule transition. So it condenses because of this polar activity only. These are all neutral polymer. There is nothing else except these activities which I am talking about, except these local forces which I am talking about. Okay. Now, but none of these model, which is ABPO or these polar active polymer, is sufficient to talk about these Janus chains that I had showed you in the beginning, right. So these Janus chains are the chains where you have each chemically active particles which are stitched to each other, and we don't know what kind of forces are actually present there, whether it's an active Brownian, whether it will fall under the category of active Brownian or the polar, we don't know that, right. So therefore we came up with a more explicit model to talk about these Janus chains, right. So these are the polymers, which is the Janus particles which are attached to each other. So you can see these dimers that I have here are the Janus particles which are attached to each other by some means, by some potentials, right. So individually, if I look at each of these Janus, you have a catalytic site where the reaction goes on. A fuel is getting converted to the product, right, A is getting converted to B. So there is a gradient of the product that I am showing here, which is formed across each Janus particle, right. And therefore, because these are the model where I have the explicit solvent into it, right, therefore I can also see what is happening to the flow around the particle, and you see there is a flow of the solvent around the particle, which we can really characterize very well, right. Now, if I have multiple of these Janus particles stitched together, what is going to happen is the question that we are going to ask, right. One thing which is very clear is for individual Janus particle, we have a gradient which is set up, and therefore this gradient will give a local force to each of these Janus particles, right. So each of these active particles is going to have a local force which is represented by these black arrows. But remember, these are chemical gradient that can diffuse across, right. So therefore, and this is the gradient of same B particle. Every genus will have the same reaction which is going on, right? Therefore, a genus which is sitting somewhere here will also sense the gradient generated by this, right? So overall, it's not a polar active polymer. Rather, it is a polymer where it has some local forces because of the catalytic, this catalytic site present next to it, and also this long range forces, right? Because of all other genus particles present into it, right? So I have now the forcing, which can be decomposed into two parts, the local force and a global force, and then we will try to see what is happening because of these forcing. So basically what is happening is some reaction on one site, right? And there are multiple sites where the reaction is happening, and because of this reaction, forces are getting generated, right? And forces are of two types. One is the local forces generated because of its own reaction, and the long range force generated because of all others, right? Now if I have something like this, and I characterize this, so we do a explicit solvent simulation. I'll tell you why we are emphasizing that also here, okay? And we see what is the structure that we form. That's the first thing that we are going to talk about, the structure. So when we have the passive polymer, you see as the time evolves, there's nothing much happens in the, as far as the structural transition is concerned. The polymer stays in a nice, because of flexible polymer, it is going to stay in the coiled state all the time. But the moment we have activity, the polymer has this coil to globule transition. So it condenses this activity that it has, it condenses the polymer into a globule, right? And then we went ahead and talked about what kind of globule it is, how do we characterize, and of course to characterize the globule, the easiest thing is to talk about the radius of gyration, and you see this is the radius of gyration. As we increase the Peclet, which is the activity on the system, it systematically goes to a lower and lower value of the Rg. That means it systematically condenses to a globule state as I am increasing the Rg, right? The next thing that one would talk about in such cases, that how compact these globules are. When you are increasing the activity, what is happening to the Flory exponent, which tells you how compact these globules are, right? So in the coiled state, if you talk about how do we get the Flory exponent, we get the Flory exponent by talking about how Rg scales with the number of monomers, right? For a passive system, it is known that it will scale as roughly 0.6, right? And as you go to a completely globuled state, it is going to scale as 0.33. Right, so we see a very systematic change from the passive polymer, which is Peclet 1 in this particular case, which is 0.6, towards 0.33, which is a complete globulization. So as I increase the activity on the polymer, right, I am actually going from a coil to a globule state transition, and that transition is very gradual, and we can talk about how this Flory exponent changes with the Peclet number. Interestingly, though our model is very different from the polar active model that I showed you, but the scaling that we see for the Flory exponent as a function of Peclet number is very similar to what they have also observed. I'll come to the reason for that also in a moment, right? So what we have seen so far is by having these activities. Now remember, this is a simulation, this is a model where I have the explicit solvent, therefore my range for n that I can go to is limited by my computation, right? Therefore I would like to talk about this kind of polymer for a long range. If I have so I'm talking about this coil to globule transition for a smaller polymer. Now the question is, if I go to a very long polymer, will we still have the coil to globule transition, or will not have? The length of the monomer plays an important role in this particular kind of activity or not, right? But then the problem was the explicit solvent simulation, and therefore we actually now mapped our explicit solvent simulation to an implicit solvent simulation. It gives me two advantages. The first advantage is I can definitely go to a very long polymer size and see what extra it brings in. Secondly, I can characterize the effect of the hydrodynamics, right? In my first simulation is where I had the explicit solvent, therefore I had the explicit hydrodynamics. Hydrodynamics is flow, what is happening because of the flow, right? Now I don't have it, so I will actually be able to characterize that what is it that the hydrodynamics is bringing to the condensation. Whether the condensation will happen or will not happen, it is because of the hydrodynamics, and so on and so forth, right? So to do that mapping, we need to figure out the forces, right? If I want to map a model A to a model 2 different simulation method and I want to compare them, I need to have the parameters right. To get that parameter right, we need to really talk about the forces which was, what is the origin of the activity in the previous case, right? I told you the chemical gradient, right, which was generated by the chemical reaction on all the genus is the reason for the activity, that is the reason for the forces, right? That is what we call as a diffuser phoretic force. A phoretic force because of the concentration gradient, therefore diffuser phoretic force, right? So how do I model these diffuser phoretic force in this particular case of implicit solvent? Explicit solvent, I can actually carry out the reaction. Right? We had the reaction, that reaction will diffuse the solvent particle, everything is inbuilt into it. Now if I want to bring out this diffuser phoretic forces, which is same as what we had in the explicit solvent, I need to get this thing right. And to do that, we employ this kind of force, which depends on the chemical concentration gradient. C is the concentration of the product particle, right? And then that depends on, so each of these source particle, right, or the catalytic particle will be the center for the chemical generation, right? And therefore each of these phoretic particle will get a force, right, which will be dependent on the concentration gradient generated by all other sum of all these things, which will come here, and then I have a parameter alpha here to play with, which I will play with and actually match these two simulation method accurately to be able to say anything about the hydrodynamic effect very, very, very concretely, right? And we do that. We talk about the Peclet number. So basically what we would like to do is we would like to get the same Peclet number from both the simulations. To do that, we have to talk about the translational velocity and rotational diffusion coefficient of the polymer, right? We do that, we see what is the value of alpha C, where we have the exact matching from HI minus simulations and HI plus simulations. So I have a model where there is no hydrodynamics, I have a model where there is hydrodynamics. I tune these parameters to get exact matching of V and exact matching of dr, and therefore whenever I say that Peclet 4 for model A is the same Peclet that I am talking about in the model B, and therefore whatever I will get is going to be same, and the extra effect that I am going to have is due to the hydrodynamics that I can say very, very, very confidently, right? So what is it that hydrodynamics is bringing to me? So you see that this is for the passive polymer, this is for the active polymer. You see for both these cases, let's talk about the active polymer because of the time. So if I see this active polymer, this is something, the blue curve is something which is HI minus, right, and the red curve is something which is HI plus. HI plus is something that I had already shown you. So you see red curve to red curve here, when you increase the activity from, when you go from passive to active, there is a condensation, Rg goes from roughly 15 to 5, so there is a condensation, which is what I showed you, right? And that condensation also happens in the implicit solvent simulation technique, but now the compactness that you have for the HI minus case is something which is even more than the HI plus case, which is also expected, right? When you will have hydrodynamics, when you will have the solvent in between, it is going to swell the polymer slightly more than what it is going to be without the solvent in between, right? And therefore we show that these are something, both of these models is something which is going to show us the condensation, the coil to globule transition. The difference in that, in the structure of that, is due to the solvent present in this. So you can see this is more open structure, but this is much more collapsed structure. But that's not what I started with. So this is one of the agenda point, that what is it that hydrodynamics is doing. So we saw that hydrodynamics is actually, and of course the dynamics part is something which I am not talking about, is also getting altered a bit. But at the end of the day, we are getting the coil to globule transition in both these cases with subtle differences. The next question that I had asked in the beginning is, what will happen if I have the long polymer, which I could not access with the explicit solvent, right? So you can see this again, the red ones are with the hydrodynamics, the blue ones are without hydrodynamics. I can access large N. Interestingly, when I go to large N, we see a non-monotonic behavior of Rg with N. That's very interesting. You see, of course, the 2 points to note here is passive will have a scaling of 0.6, but when I go to active case, the scaling decreases from 0.6 to roughly 0.3. That shows a globule formation from a coiled open state to a closed state formation. That's one thing. The second thing, important thing to notice is the Rg increases, then decreases, and then again increases with an exponent which is roughly 0.33 again. Now the question is why this is, and why do we have a non-monotonicity? Why don't we have so we have 0.33 here, 0.33 here, but in between there is a dip in Rg, right? And we feel that this is due to the competition between the activity and entropy. So when you increase the size of the polymer, the entropy of the system increases. At the same time, when I'm increasing the size of the polymer, number of active sites are also increasing, right? So at long time, this gets a equilibrium, but in between, where the entropy is, so the active forces dominates the entropy, you get more collapsed than the short time and the long time. Right? So that's the reason for non-monotonic behavior that we observe in this particular case, which we couldn't have done with the explicit solvent simulation. Now again, coming back to the same in the initial thing which I told you in the beginning, that these polymers, so I'm going to go to the ring a bit, but you imagine the same thing is going to happen to the linear polymer. Remember, I said we have two types of forces in these chain of Janus. A local force because of the catalyst, because of the chemical activity of the adjacent particle, and a long range force, a global force, right? Which I could not really differentiate in the previous simulations, right, in the previous work of explicit solvent. Now I have this model where I can actually differentiate these two forces very well and try to figure out what is it which is important at what length scales, right? Is it that the local force which is important, or it's the global force which is important in the condensation of the polymer, right? So polymer is going from coiled state to a globule state. There are two types of forces, the local force and the long range force. Which of these two dominates when? Is it one of them always dominate or the other one dominates? These are the questions that we start asking now. So we have now these two forces explicitly written, right? And I can control these forces because I have now this implicit solvent completely nicely mapped to the previous one, right? And then I go ahead and talk about these. Okay, how much time I have? Oh my God. Okay, so that's cruel. Let me just go back here. Okay. So you see here, we have this orange curve where we have both these forces. In fact, in all of them, we have both these forces present. The local force, which is PT, and a global force, which is PR. So we see that there is a non-monotonic behavior that we see for because of the presence of both of them. Now the question was, this non-monotonic behavior that we observe is because of which force, right? And therefore now we simulated this polymer for the PT local force separately and the global force separately. What we see is this non-monotonic behavior that we observe is just because of the local force, not because of the global force, which is not very intuitive, right? Intuitively you will think that it's the global force which is trying to bring things together, which has to dominate, right? But that's not what we see. In fact, the next slide is something that is that's showing us the same thing, that for a long polymer and even for the short polymer, it's the local force that dominates, and that is the reason for it to condense, not the global force, right? Now the question is why? Why would I think that local force is something which is going to bring the monomers together and condense it? The answer to that lies in the bond-bond correlation. If you talk about if you look at the bond-bond correlation along the polymer, you will see a hairpin sort of structure forming when you have the local forces. That doesn't form when you have the global forces. So what happens is, first of all, when you have both these forces present, the local force, the local tangential force, which is important for the condensation. And why does that happen? Because these kind of local hairpin-like structure, multiple hairpin-like structure that forms, and that is what gives rise to this kind of condensation. Okay, because of lack of time, I'll skip this, and just the last part that I wanted to talk to you about is Now that we have understood that these local forces are important in condensation, so let's forget about the global forces and try to see what is the effect of these local forces. If I change the density of these local forces, density of these motors, what is it that it does? So when we change the density of the motor, we see a systematic change in the conformation. When you have multiple motors, it goes to a condensed state. The moment you decrease the number of motor, it goes to an open state. And that how much it is going to go to an open state will also depend on what petle we are talking about. For a smaller petle, even a smaller number of motors is sufficient to make it take it to the collapse state. Not only that, self-similarity is lost and all that, but this is one last slide which I wanted to emphasize. Not only how many motors, but where they are positioned is extremely important, right? So we're talking about now the polar forces, which is the kind of forces you can think about in the chromatin also, simple force sliding type of protein which is recruited, which is going to give a local forces, right? Where those motors are, how many of them are extremely important. If I have a uniform distribution, this is the solid curve that you see here. If I have the same number density of motor and a uniform distribution, it shows an open state, but the moment I make the distribution of the motor random with the same motor density, it goes to a very mixed state. It's a plethora of conformation that it takes. Not only that, where the head motor is lying, right, the first polar motor, where does it lie is also extremely important. You see, here the first motor lies actually at the head, but here the first motor lies after 4 or 5 beats, right? So the moment you shift the leader to the back, the passive ones dominates and they start disorienting the whole thing, and it goes to a collapse state. So from a very nice open state to a collapse state, but just by shifting the motor position by 3 or 4 is sufficient to take it to do these transition, right? So with that, maybe in the interest of time, I'll stop here and thank all the students who have done. The major part of this was actually done by Sandeep and Surabhi, and of course this is taken up by these 2 kids right now. So thank you all, and I'll be happy to take some questions.
Thank you, Smita. We can take a couple of questions. Yeah.
Very interesting. I had a comment, it's not so much of a question. Do you think that if one thinks of a polypeptide chain as an active polymer, then one can describe well-folded globular single domain proteins as things that have high motor density, and IDRs and IDPs as something that have poor motor density, and the metamorphic proteins as some things which,
I would love to
which have random distribution of motors or
Absolutely.
similar motor density, but
Absolutely.
a plethora of conformation.
Absolutely. This is basically the main slide which I wanted to say. So actually this polar active polymer which we are talking about is something that can be very much related to these the different types of conformations that we observe, and how many motors are there, where they are situated. These are something which are extremely important to find this.
Folding cores or
So what I will do is, so the nice folding structure that we get in the folded the peptides is something that we have not really seen here, because there's a lot more complexity that needs to be brought in. First of all, these are all flexible polymer. If I bring in the semi-flexibility, things will change, right? So, and then maybe the folding will change, and whether we are talking about linear or ring, all those things changes the folding structure a lot. Right, so I'm
But IDRs and IDPs are low complexity regions. So
Right.
you can postulate that therefore they're not as active as polymers as folded proteins.
correct. Yeah, we can say that. We can say that. We can very well say that. That's the hypothesis that I would like to bring in this case.
Sandeep, question?
So, again, I mean, this is something perhaps we also discussed earlier, but there are different classes of these active polymer molecules.
Absolutely.
Now, is there some sort of a universality that you find? I mean, of course, you talked about some interesting behavior, but is there any kind of unifying principle? Like if I go back to critical phenomena, right? There you have beautiful like exponents and so on that do not depend on the actual form of activity, but say that there are very different ways to actually include activity in your model. Now, is there a way to actually somehow unify them?
So far, no. So far, no. People have been trying, and there are again, people are coming up with different types of activity, right? For example, this ABPOs, polar active, Vishik model for this thing, right? These are different types of activities, but there is no unifying picture that has emerged so far in the active polymer this thing. But yeah, one thing is which is there in all of them is this condensation. Whether you talk about the Vishik activity or you talk about polar activity or ABPOs, this condensate formation is something which is there, but scaling and all those things are very different right now.
We'll
And
take
it's-
one
it
more
has to
question
come.
before we break for coffee.
So, just one thing I just want to understand is that your concentration gradient, what is the length scale?
It's a long range concentrate because it diffuses.
Diffuses,
Right,
right?
it's a-
So it's long range. Now, when your reaction is happening, then to set up the concentration gradient, there is also
Time
also
scale.
a long time scale
The
problem.
time scale.
And your change of the beat, beat is also changing its position. How are you managing all these time scales? That's one thing I'm just
So one thing that I will tell you that all these characterization that we have talked about is in the steady state. So I have let the system evolve to this whole concentration gradient, whatever it can, the maximum product that it can form and the diffusion and everything. And all these studies that I have talked about is the steady state. But again, there is something that we can talk about the dynamics, how it goes. Those results are something that we have done and I have not really presented in that
just
case.
Another point, let's say start with the very length polymer which is which is coiled, and it goes to a globular state. What kind of phase transition are you really can you characterize this transition to any order?
Okay, it's something which is very non-trivial, I don't think.
You cannot define a free energy.
Exactly, you can't define a free energy, so it's very hard, yeah.
Is it kind of at least cooperative or not? That's the point I'm trying.
Yeah,
Not-
so-
not in the order.
Yeah, something which, see, it has to be some sort of a cooperativity has to come in to bring in this kind of globalization. Otherwise, if it is non-cooperative, things will not really come to this condensed state. But then order transition is something that nobody has, anyone cannot define.
Cooperativity probably still can be defined. How
So that's something which is, I think for me also it's an ongoing work, and if I will meet you next time, I will hopefully try to tell you that.
I still see a couple of hands at the back, but I apologize. We have a coffee break now. That's the great thing. So you can catch up with Stigma and get your questions solved. I request all of you to reconvene in 20 minutes so that we can start the next session on time. We'll still be late, but not too late. So let's again a big round of applause for the two speakers so far. All right. Once again, welcome all of you for the last part of the meeting. This is going to be Punaran Gupta Sarma from ISEB Mohali, and he'll be talking to us about phase separation of bacterial genomes and biofilm matrices, experimental observations, and some hypotheses. So over to Punaran.
Thank you. Thanks, Prashad. And many thanks to Divan and Sandeep for the invitation. I've had an incredible 2 days. The sheer diversity of presentations has been really enjoyable. So I'm going to talk about the phase separation work that we've been doing for a few years now, but I must warn you that this is a recent interest. I have actually, over the last 28 years or so that I've been a PI, I've been working on 8 or 9 different things, some of which I continue to work on. Currently there are 5 things going on in the lab. Phase separation is one of them. It's a recent interest, started around 2017 or 18. And somewhere in the middle of the talk, I'm going to use some stuff that I've written here. It's not very visible, but I'll read it out, right, when we come to that. But please don't get distracted by that. Let me go into the talk and ask you to look at the slides instead. So as Purushad said, I'm going to talk about phase separation of 2 kinds. One is the genomes that are present within bacteria, and the other is the matrix that exists outside bacteria when bacteria form something called a biofilm. And so this is a standard schematic of an E. coli bacterium, and you can see that the DNA is actually occupying a very small fraction of the cell's volume. The rest of it is cytoplasm, right, and then the cell membrane, the cell wall, and so on. So there are a few moot questions that arise. The main question is that the genome of a bacterium like E. coli is 6.4 million base pairs long, right, and at approximately 3.4 nanometers per base pair, that's about 2070 odd microns. So how does something that is 2070 odd microns get packed inside a cell that is less than a micron in length and less than half a micron in diameter, right? So there has to be a huge amount of compaction, which is about a 2,000-fold compaction that needs to happen. And the problem here is that double helical bDNA is like a car's speedometer's rigid torsion cable, largely because of charge-charge repulsions among phosphates, but also for other reasons. And DNA is very difficult to bend, loop, or bunch due to this, right? And there have been talks today, including one this morning, where we saw DNA, being very rigid. So people have suggested over the years that supercoiling and bending of DNA by proteins do explain a certain level of compaction, but they certainly don't explain the 2,000-fold level of compaction. And more importantly, they don't explain the perpetuation of that compacted functional genome over hundreds of cell generations, because remember, the DNA cannot exist in bDNA form, in an open form inside the E. coli. It will just tear through the membrane, right? It's 2,000 times longer than the length of the E. coli cell. So it has to remain compacted and then do all its transactions, replication, transcription, everything while it is in that compacted state. So what is it that explains the perpetuation of that compacted state is the question. Is there a genetic mechanism that keeps bacterial DNA constantly compacted? And the question that we're asking is it liquid-liquid phase separation, which is mediated through mutual macromolecular crowding of DNA by protein and protein by DNA, and derigidification of the DNA through charge neutralization by positively charged proteins that bind to the phosphates on the surface of DNA, but in particular multivalent proteins, multimeric proteins, and non-specific, non-sequence specific proteins. So let's jump to an experiment straight away. So to check whether what we have proposed in the previous slide is true, we added HUB, which is a histone-like protein for bacteria, to salmon testis DNA, and we found evidence that there is liquid-liquid phase separation as well as compaction in a graphic video taken on a non-confocal microscope here, which I'm going to show you right now. So this, watch this lower left panel. this is basically a lot of DNA which is lit up by a dye that intercalates within the DNA, cytogreen. And now what we are doing is, this is just a drop lying on a slide, what we are doing is just taking a little pipette and introducing some HUB. And of course the introduction causes a vortex in the liquid, so you will find things are rotating for a while, but look at what happens. So here we introduce the DNA, the HUB, and immediately there is compaction. And as this goes round, you can see that the DNA, which was otherwise uniformly dispersed over the drop, is now clumping up, right? Can you see the clumps as they go around in that vortex, right? It's clumping up, and then if you just wait for a few minutes, this turns itself into this beautiful dispersion of liquid-liquid phase-separated droplets, where basically what is happening is HUB is a dimeric molecule which also tetramerizes and then octamerizes, and each dimer has 4 DNA binding sites, 2 canonical sites and 2 non-canonical sites that our group actually confirmed. Someone else had proposed it. So it has 4 sites for DNA binding per dimer. So if a fraction of the HUB population is octameric, that means it has many sites at which to bind to DNA. So what it does is, wherever it binds to DNA through charge-charge neutralization, it causes DNA to de-rigidify, or what we would say in Hindi, pilpilaya jana, the DNA softens. And then once that happens, because the HUB is a multimer, it grabs the DNA here, there, and everywhere, and bridges and bunches it very much like what we heard today morning, in regard to the protein BAF. And so causes the DNA to get crowded and allows those phosphates to approach each other, because now they've been neutralized through the arginine and lysine residues that are on the surface of the HUB. And that macromolecular crowding of the protein by the DNA and the DNA by the protein, each of which you can independently get done through the excluded volume effect that polyethylene glycol exerts, right? Where you occupy or distract the solvent with a lot of hydrogen bonding with polyethylene glycol, and so you have very little left over for the protein, and that crowds the protein into a small volume. Something very similar is being done here through mutual molecular crowding by DNA and protein. And so this is not an intrinsically disordered protein. It's a very structured protein, as you can see. It has helical regions. It also has beta sheet regions, which are part of the canonical DNA binding motif. But there is a little bit of IDR also in there, which you obviously can't see in a crystal structure because there's no diffraction, so the signal doesn't build up, so you won't see it in a crystal structure. Okay, so we said, hey, if HUB can cause liquid-liquid phase separation of DNA, and that's salmontistis DNA, how about checking it within the E. coli cell? So we created RFP-fused HUA and also venous-fused HUB, and then we looked at the cells. And one thing that I must tell you about HU overexpressing cells, for a reason that will become evident if I have time during the middle of the talk, is that these cells don't very easily dissociate after cell division, so they form these long filaments in which cells have divided, but they remain connected. So these long filaments, like you can see here, right, some of these long filaments. So this is one long filament, and what you can see here is individual E. coli cells and their genomes. The first thing that strikes you is that each of these looks very different. Each genome in this string of E. coli cells looks very different. And then any one of these, if you watch over time using super resolution microscopy, which is what I'll show you in the next video, can you see that each genome is also changing shape, right? And what we could see in this and in a bunch of other such images and videos is that it seemed as if there are these very small submicron sized liquid-liquid phase separated droplets that are fusing and separating, perhaps as transactional processes are happening on the DNA, replication, transcription, and what have you. So what might be happening is that the E. coli genome, which is circular, is like a string of beads. Each bead is a liquid-liquid phase separated droplet, and sometimes they come together and fuse, and sometimes they separate depending on what the polymerases are doing, right? And so this is actually a time series of one of these, or a few of these, one of these chromosomes. You can see that they are changing shape over time, right? So they look like they are basically submicron sized liquid droplets. And if you do it in vitro, so the first one was with salmon testis DNA and cybergreen, this was with RFP labeled HUA, and what you have here is a fluorescently labeled form of HUB. So we've used a variety of fluors over the course of this study. Some of them are Alexa fluors and so on. So you can see that these very nice droplets do surface wetting when you compress them. They show dripping behavior over time as they age. The little pieces that drop off and they lose their sphericality. And in addition, they also show fusion. Yeah, there you go. So one bit coming there and then colliding, and then having some trouble initially at first interaction, but then eventually fusing, right? So fusion is observed. So clearly showing that these are LLPS, right? And also FRAP of one of these droplets, bleaching recovery, suggests that there's a diffusion coefficient here in meter square per second, which clearly shows that this is liquid. Now I'm going to rush through some of the remaining slides because I've made the main point that I want to make in this section of the talk. So what I'm showing you here in this slide is basically 50 micromolar HUB. Now I'll explain why 50 micromolar in a second. Labeled with Alexa fluor on the left panel, and 4-way junction DNA, so not salmon testis DNA, but a cruciform DNA that we make using 4 synthetic oligos that come together to form a cruciform. 4-way, and then for most of the rest of the study, what we used is that 4-way junction DNA. So you can see the DNA here labeled with DAPI. You can see the HUB labeled with the Alexa fluor, and when you merge them, you find that they're absolutely, coincident, co-localized. So every bit of the droplet contains both 4-way junction DNA and HUB. And condensates are formed only in the complete, condensates here notably are formed in the complete absence of polyethylene glycol. So this is absolutely natural physiological conditions, pH neutral, and using DNA concentrations of 4-way junction DNA, for which the base pair concentration is actually subphysiological, and using an HUB concentration which is physiological, right? You can, from a knowledge of how many HUB molecules are there in the cell, you can calculate what the actual concentration is, right? So without PEG, you get condensates under entirely natural physiological conditions of pH, ionic strength, and everything else, 150 millimolar sodium or potassium chloride. And without PEG, there are no protein only or DNA only condensates observed. There are only protein DNA condensates when you add them to each other. So we looked at the CSAT values, which this bit of it is expanded here, and you can see that it's of the order of about just under 2 micromolar. Now we also looked at HUA, which showed a CSAT value that was 10 times higher, and we have some reasons for that. But for the moment, this is still work on HUB, where you can see that higher DNA concentrations, when you increase the micromolarity of the 4-way junction DNA, you see that now turbidity here is a proxy for LLPS occurrence, which has already been established. So you see that higher DNA concentrations achieve more condensation, promote condensation. Higher HU concentrations for a fixed DNA concentration also do the same thing, right? And that's the control without the DNA. And a higher temperature also causes greater condensation, but higher hydrophobicity does not in this particular case. And the reason we feel is because it's largely driven by electrostatic interactions. If you break electrostatic interactions, there's clear evidence that you discourage condensate formation. So both with increasing concentration of a salt like potassium chloride and with increasing pH, you find that turbidity drops precipitously, shown here by representative microscopic micrographs. You can see that basically it's the electrostatic interaction between, non-specific electrostatic interaction between HU and DNA, which is causing them to mutually crowd each other and cause droplets to form. And if you break those electrostatic interactions with salt, then you break the condensate formation. But there's another reason that this can occur, and that is that at very high salt, the salt can actually distract the solvent, because in order to be soluble, a large part of the solvent may go away and solubilize the salt, and so there may not be no water available for the protein. That can also cause some reentrant behavior, presumably at high concentrations and so on. Okay. Now, this slide essentially says that HUA and HUB have related sequences, very similar structures. So here you see a superimposition of one in green and one in pink. You can see that HUA and HUB structures are virtually identical. And if you have a heterodimer, so usually they form homodimers, HUA with HUA, HUB with HUB, but when you mix them, a large part of the population goes to form heterodimers. So this is a heterodimer of HUA, HUB from a crystal structure that is known. So we made pure homodimers of HUA and HUB. And the data with HUB has already been shown, so this is data with HUA, where you see that the CSAT is not 2 but over 20 micromolar, and you get relatively tinier droplets, which also, however, show you full recovery of fluorescence after photobleaching. So HUA appears to be poorer. And the reason is that this is something that has been talked about in the literature by a few other people, but we are actually showing it here with some data, both by acidic native polyacrylamide gel electrophorese and also by gel filtration or gel permeation chromatography, that HUA is a pure dimer and does not form bigger multimers, but HUB is a mixture of dimer, tetramer, octamer, and other species, right? So being more multimeric, it's more multivalent, and therefore it achieves a greater amount of condensation because it can pull in the DNA to a greater degree. So what we are showing here is there are a couple of mutations that someone else called Shankaradya found, which can turn HUA into almost an HUB mimic. By making two mutations, you can cause HUA to form octamers instead of dimers. And so we made those mutations, and you can see here that this is the behavior of HUA as a function of protein concentration. This is the behavior of HUB, and this is the behavior of HUA with two mutations which cause it to behave a little bit like HUB. You can see that also in these micrographs, where you can see that the mutant actually begins to show LLPS at much lower protein concentrations. This slide shows you that HUB and HUA coacervate, presumably because when you add them to each other, a lot of the population forms heterodimers, and so they form these complex coacervates. However, here you can see in the same coacervates, this is HUA labeled, that's HUB labeled, the merger is almost perfect, right? And these are the Pearson correlation coefficients for the colocalization, which show you that the colocalization is near perfect, right? And they've almost reached a value of 1. This shows you something interesting which we did not anticipate. So when you add a little bit of HUB to a lot of HUA, you actually get synergistic behavior, which is best shown here by this one thing. This 20 micromolar HUA achieves this much LLPS, 5 micromolar HUB achieves that much, but when you add 20 micromolar HUA and 5 micromolar HUB, you get a whole lot more because there is this synergistic behavior between HUA and HUB. Okay. We also created a HUA-HUB heterodimer proxy by fusing HUA and HUB genes to create a forced heterodimer formation, and those also form condensates with four-way junction DNA, and that's the behavior that you see with protein concentration. We show that multiple forms of DNA can be assimilated by HUB into the same niche. So this is with double-stranded DNA, with four-way junction DNA, Nick DNA, and so on, right? And in all cases, you get different efficiencies of this happening. And this might be the last slide in this series, which shows you that these condensates can also incorporate DNA polymerase. So you let in DNA polymerase. Now DNA polymerase has affinity for DNA, and there's DNA inside the droplets much more than there is DNA outside the droplets. So the polymerase gets titrated into the droplet, right, just to show that it's possible to conceive of replication, transcription, and so on happening inside the droplet, right? So to summarize this, HU and DNA cause each other to undergo LLPS. The LLPS leads to compaction of the genome into the nucleoid, and the LLPS is the generic mechanism by which it is possible to compact DNA into those small spaces. And this nucleoid persists and is constantly remodeled, right? Therefore, LLPS is the primary generic mechanism for compaction of DNA in the bacterial genome. Now, the question is HU the only non-sequence specific nucleoid associated protein that's there in the nucleoid? Largely yes. During bacterial growth, HUA is what dominates, but when you go from a growing phase to a stationary phase, then the HUA gets degraded, and in its place another protein is produced at much larger levels called DPS, or DNA binding protein from starved cells. So this is from an old dated paper showing 12 nucleoid associated proteins. Later this has been changed, but nobody's drawn another diagram. So 5 of these have gone off the list, and now there are 7 nucleoid associated proteins. 2 of them are non-sequence specific, HU and, well, HUA and HUB, 2 isoforms, and DPS, right? Now look at what happens here. So in the early exponential phase, HU dominates, right? And then in the late stationary phase it goes down. Instead, there's hardly any DPS in the early stationary phase, but in the early exponential phase, but in the early stationary phase and late stationary phase, while HU drops, there is a concomitant increase to almost 180,000 copies of DPS. It's the single largest expressed protein in E. coli, right? And this is what its structure looks like. That's a single monomer of the dodecamer. And when you express it and go to the late stationary phase, you actually get crystals of HU forming inside the E. coli cytoplasm, which seem to wrap up DNA, not like the histone octamer, in a very different way though. Okay. So the question is, if HU does it, then what about DPS? So we found that DPS DNA solutions also turn turbid, just like HU. We found that DPS can also create condensates, but they are micron sized and submicron sized, whereas HU condensates are 4 to 5 microns in diameter, and some of them are aberrantly shaped, as you can see here, right? So these seem to be conglomerates of spherical submicron sized droplets that seem to come together. So it's very non-classical, non-canonical behavior for a protein DNA condensate. And of course, if you just use 10% PEG also, you can create beautiful condensates of DPS, but with DNA, it forms these non-classical condensates, right? And these condensates also show rapid recovery of fluorescence after photo bleaching. And when you now add all 3, HUA, HUB, and DPS and DNA, all labeled by 4 different fluorophores, and then do multi-channel imaging, then you can see that the HUB, here in this particular case, you're not seeing the HUA, the HUB and the DPS co-localize, but when you zoom in, you can see that there are these, heterotypic coacervates within coacervates that are formed, right, which you can see here, which are basically DPS-rich, DNA-rich regions within DPS-poor regions that are, however, still HU-rich and DNA-rich. Right? And so if you look at them in 3 dimensions, then that's what they look like. These yellow bits are those DPS-rich regions. So this suggests that they can coexist in the early stationary phase when both HU and DPS are there on the genome, in different parts of the genome. But interestingly, I do not mean to suggest that where there is DPS, there is no HU, or that where there is HU, there is no DPS. Everything is there everywhere. It's just that DPS dominates the DPS-rich regions and HU dominates the HU-rich regions, but they separate out, right? Okay, so these are, so this is another slide that shows that DPS is enriched in the DPS phase in those multiphasic condensates. And we also found that in some experiments, upon overnight incubation of HU, A and DPS with salmon testis DNA, not 4-way junction DNA in this case, you get these very natural, natural is this chips, right? Shaped crystals. You can see them here and here, right? you get these natural shaped crystals, which are probably tetrahedral crystals, but they just happen to be lying in such a fashion that they look triangular, right, to the eyes. And which also show liquidity. See, these are not spherical, but you bleach them, and then you do get a little bit of recovery, but it's not a whole lot because it's more crystalline than liquid-liquid phase separated. So we've talked about the bacterial genome being compacted by HU and its two isoforms, or by DPS, or some combination of both. Yeah. And so now I'm going to talk about the what's outside the cell, right? So, so interestingly, speculation about HU and DPS somehow being involved in the nuclear phase separation has existed. So here's a paper which says, while the behavior of DPS and HU on bacterial genomes is consistent with the role in phase separation in nuclear organization, neither DPS nor HU have been demonstrated to phase separated either by themselves or with DNA. So that was the little bit that we did there. I'll skip this slide. I want to talk about it. Okay, so all of this work appeared a little less than 2 years ago in a paper in JBC, where at one point we also roped in my colleague Samrat. So the next point, which I'll make very fast, is that DPS, HU and DNA are not just there inside the cell. When a biofilm forms, the main constituent of the biofilm, which is the extracellular matrix, is basically extracellular DNA. Okay? So eDNA is a key component of the biofilm matrix of most bacteria. This began to be realized around the year 2015. And how does that eDNA come out? Obviously it comes out from within the cells, right? So when cells are growing, although they all have negatively charged surfaces, they tend to stick to each other because in a separate paper a year before that in JBC, we showed that HU does not just exist inside the cell, it also exists outside the cell on the surface of the cell, bound to lipopolysaccharide. So lipopolysaccharide is negatively charged, HU is positively charged, and HU binds to lipopolysaccharide because lipopolysaccharide has a head group which has a sugar phosphate arrangement that is not dissimilar to the sugar phosphate backbone chemistry, right? There are two sugars there, they are hexose sugars, not pentose sugars, there are two phosphates there. So we showed that HU uses its DNA binding regions to bind to lipopolysaccharide, and that therefore it acts as a glue in biofilms, because biofilms have this huge amount of DNA that's been thrown out through explosive cell lysis, but that DNA is coated with HU. So the surface of the E. coli is negatively charged because there are phosphates on the lipopolysaccharide. The surface of DNA is negatively charged, but the E. coli binds to the DNA through a glue which is positively charged and which is multimeric. So you have a multimer of HU which binds to the cell on the one hand and binds to DNA on the other hand. So it neutralizes charges on both, right? And so this ability of HU does not need to involve DNA. We've also shown that HU causes cell-cell clumping because HU can bind to just LPS here or LPS there. LPS here, DNA there, DNA here, DNA there, all possibilities, right? Using the exact same sites. So what happens is that these cells, as they're growing, they tug at each other, and then suddenly one cell in the population will become spherical because its cell wall would have broken due to physical pulls and pushes, right? Because of the tugging, because of the HU trying to attach cells to other cells, and then suddenly that cell explodes and releases its DNA. And so in a growing population of cells, if you put in some DNA binding dye, you will find that there are flashes of DNA, and ultimately there's a huge amount of extracellular DNA released by cell lysis, and that DNA is also HU rich, right? So we asked, if extracellular DNA is the main component of biofilms, then biofilms should also be phase separated, and I'll not give you the whole story here, but I'll just, yeah, so by the way, this was a paper that we published which shows that HU is a molecular glue that attaches bacteria to extracellular DNA in biofilms. It's a very detailed study that has also been taken up by other groups. Now, what we did here was these are HU DNA condensates of the variety that you saw in the first few slides. And we said, well, if there's DNA inside and I add E. coli, the E. coli has lipopolysaccharide on the surface, that should bind to the HU that's in the condensate. And right, so therefore, the E. coli cells should titrate into the condensate or onto the surface at least in the beginning. And we actually have evidence for that. So these E. coli are GFP expressing E. coli. When they're added to these red HU DNA condensates, they all get titrated to the surface of these condensates. You can see that the medium is depleted, everything is on the cells, there's perfect co-localization. And here are some interesting pictures showing that there are E. coli cells that have also entered condensates, like a proxy for biofilms. So basically, this is a biofilm of DNA with HU, and then later we've added DPS also, and these are fluorescent E. coli which are co-localized inside these, suggesting that not only are E. coli cells compatible with the LLPS, which is the bacterial biofilm, but the surface of the E. coli is also a two-dimensional LLPS, because HU or DPS bind to the lipopolysaccharide on the surface of E. coli. So that's summarized. Anyway, part of that work was shown in another recent paper, which was this one in the Journal of Bacteriology. E. coli cells advance into phase-separated biofilm simulating extracellular polymeric substance containing DNA, HU, and lipopolysaccharide. Now, interestingly, what we found was that if you have HU lipopolysaccharide condensates, which also form, by the way, as you see here, HU LPS, right? But they don't show fluorescence recovery after photobleaching, whereas HU DNA does. And when you have a mixture, then you get intermediate behavior when you have HU LPS and DNA. Okay, so this is the summary that I'll leave you with. That we showed that DPS also binds to LPS. So there are basically four players here. There's HU and DPS, which are DNA binding proteins, and there's DNA and LPS, which is a proxy for DNA. And in every combination, they can bind to each other, and they all cooperate to form both the phase separation inside the nucleoid and also the phase separation in the biofilm. I think. Okay. If there are any questions, I'll deal with this last slide, which is a very exciting recent thing. In the meantime, can I just take half a minute to say what I wrote on the blackboard? Okay. So here I've written what happens upon crowding. You could come to this side, Devan. What happens upon crowding, right? So these rectangular boxes are schematic representations of protein. These are the water molecules that are bound to the surface as the bound water, which is usually a single layer of water on the surface. And these are bulk water, isotropic, right? And so this is the situation when molecules are not crowded together. But when molecules are crowded together and they are so close to each other, pushed into close proximity, where there are only maybe 3, 4, 5, 7 layers of water between the one protein and another protein, or one DNA and another DNA, or one protein and DNA, or whatever, any kind of macromolecule that can bind to water through hydrogen bonding. Then what happens, we think, is that the bound waters, through their electric fields exerted by the dipoles of those bound waters, cause the bulk water in between to also align, such that inside the droplet, the solvent is no longer isotropic but anisotropic, right? Because of the water dipole effects exerted through the electric fields, because these are actually very high field strengths. If you have water dipoles that are aligned, you can actually create a huge number of thousands of volts per millimeter of length.
Then we will not call those water molecules inside the droplet as bulk water, right?
No, but there may be some bulk water present in between too, but a small fraction. Most of that water would be immobilized water, immobilized like the water on the surface of the of whatever is undergoing molecular crowding. just protein or just DNA or a combination of, right? So this is, I think, what happens upon crowding. Basically, if the water here inside the droplet is not to exchange with the bulk water, which is isotropic, obviously it must be organized differently and unwilling to move and getting some solace or some comfort by being in this form, right? So it's not really which protein. Of course, different protein sequences, different structures will show you different kinds of behavior, but ultimately what matters is that the water inside and the water outside have to be differently organized. So the hydrogen bonding scheme has to be different, and that has to be supported by the layer of bound water on the surface of every molecule, which reorganizes the little bit of water that's between the molecules because of that close proximity and those electric field effects. So that's what I think happens upon crowding, and what causes, and this is what I think also happens in the nucleoid and in the biofilm and so on. What causes crowding? So one can be just extruded volume effect of solvent distracting substances like polyethylene glycol, right? And we know that PEG causes LLPS, but more importantly and more biologically and physiologically relevant, the mutually crowding polyvalent interactions of multimeric substances or long polymers, right, like DNA-protein interactions. So when you have DNA and protein, you don't need PEG, because DNA crowds the protein by pulling it together, and protein crowds the DNA by pulling it together. And so they kind of keep the flock together and come into close proximity, and once they come into close proximity, click, the bulk water between them turns into non-bulk water. Very much like if you have a lot of iron filings on a piece of paper and you suddenly bring a magnet, all the iron filings align, right? Because of the magnetic field effects. So something similar must be happening inside the droplet. And so then you don't need any excluded volume effect exerted by any PEG. Thank you.
Okay, let us all thank Punnanan. So if there are other questions, please. Are there any questions?
So about this last part that you're talking about this macromolecular crowding, so how does this arrangement, the different arrangement of water affect the properties of the condensate? Like what are the implications that it has